Hello viewers, in this video we are going to explain how to subculture the mammalian cells. These cells can be grown in various culture vessels like T25, 100 mm or 60 mm. Initially, you can remove the media or the spent media or utilized media from the culture vessels by using the serological pipettes, or maybe sterilizer tips can be used to remove the media. And then you need to wash the cells where you can eliminate the unwanted cells or debris or dead cells by washing with the PPS. The washing should be very gently so that cells should not dissociate easily, and then you can discard the washed PBS. And then you need to add the dissociation reagent trypsin to increase uh, the detachment of the cells. So you can use the minimal amount of trypsin for the various vessels. For example, for the T25 flask I am using here 500 microliters or less than that. It is sufficient to dissociate all the cells from the T25. So to increase the dissociation efficiency of the trypsin, the observed temperature should be 37. Hence, I am keeping the cell containing vessel inside the 37 incubator. So I'm waiting for 5 minutes. After 5 minutes, you can take it out. We can cut the cells by putting some medium to centrifuge it. Now I'm adding the medium, TMM medium. So this DMM medium containing the serum which neutralizes the trypsin activity. Hence, there is no harmful effects of the trypsin to the cells, or there is no activity of the trypsin occurs. So completely you can remove the cells, or you can collect the cells into a graduated centrifuge tube for centrifugation. After the collection of cells, you need to collect into the centrifuge tubes. So I'm taking entire cells and also trypsin containing medium. You can observe under microscope whether any cells were left over in the culture vessels to make sure that we collected entire cells and then labeled properly. So keep a balance between the tubes and then centrifuge at 1200 rpm for 3 to 4 minutes at room temperature. It's fine. These are the mammalian cells, so you should not increase the revolutions per minute more than 2000, because they don't have any cell line that will uh, degrade the cells. And then correct the cells which is in the form of pellet, discard the supernatant, and without disturbing the pellet contains the cells. Without disturbing the pellet, you can discard the supernatant completely. You can remove it, and to the pellet you can add the fresh medium. Based on your experiments, you can add into different culture vessels and also different kinds of flasks based on your experiments. So completely resuspense the cells with the medium. Here we are using DMM, high glucose medium, to grow the breast cancer cells. These breast cancer cells can be resuspended by using the DMM medium and then subcultured into different flasks based on our experiments. So here we are using 100 dishes which contains which requires 100 10 ml medium, and we completely filled up with the 10 ml medium of each plate, and then we added the resuspended cells into it. After adding the medium, the resuspended cells can be added in this. These things can be added it, and then gently you can shake or you can tap it to distribute into the plate. Then make sure that there should not be any clumps or aggregations of the cells, and they might form the aggregates if you are not disturbing this, and then label it properly. You can keep in that's an incubator at 5% humidified CO2. These are then keep it in the incubator for 6 to 8 hours to adhere it. If you like this video, please share and subscribe our channel. Thank you very much.